The overall goal of this microRNA in situ hybridization is to detect the localization of the microRNA of interest in breast cancer tissue.
So the method can answer the key question in breast cancer field, such as whether the microRNA of interest is oncomere or tumor suppressor. The main advantage of this technique is that it can detect cellular localization of the microRNA of interest in breast cancer tissue.
Begin tissue preparation by removing paraffin from previously prepared breast tissue sections. Immerse the slides into a Copland jar in fresh xylene 2 times for 10 minutes each. Rehydrate the tissue section with incubations in decreasing concentrations of ethanol in double distilled water for 5 minutes each. Next, immerse the tissue in diethyl pyrocarbonate or DEPC treated water for 5 minutes. While the tissue is immersed, make boundaries around the tissue section with a hydrophobic barrier pen. After the DEPC immersion, fix the tissue with 4% PFA for 15 minutes. Follow the fixation with a PBS wash. Then, immerse the tissue in 0.3% Triton X100 in PBS for 10 minutes. Incubate the sample with 50 micrograms per microliter of proteinase K solution for 15 minutes at 37 degrees Celsius. Then, incubate the tissue sections with triethanolamine and acetic anhydride for 5 minutes at room temperature, and follow with a PBS wash.